Thank you, David, so much for laying on this Michelin-starred feast for the mind. I don't think I could have ordered something so satisfying and delicious if I had had a menu. So thank you so much for this entire occasion. I feel extraordinarily fortunate to be here. My talk is about how the principles of neural design as set out in Sterling and Laughlin's seminal text that actually isn't cited quite as much as I think it ought to be, but that they published in 2015. And the reason I'm doing this is because I am interested in the principles of biological cognition. Now, Sterling and Laughlin don't use the word cognition, but I do. And they talk about design, they talk about behavior, they talk about information processing, but they do not talk about cognition except in cases of nervous systems and fairly well-developed ones at that. But my purpose is different from theirs because I am interested in adding my brick to the foundation of what I hope will be a future Copernican revolution in the sciences of the mind that will shift the focus of our investigations from human beings and mammals and other charismatic fauna to all of life. Now, I'm also interested in developing something that Dick Neiser pointed out in his 1976 follow-up to Cognitive Psychology, in which he pointed out that the cognitive revolution had failed to provide human beings with a view of the mind, of cognition, that they could actually relate to. He pointed to Freud, who taught us that there were hidden depths, that we had hidden depths, and even Skinner, who taught us that our behavior could be conditioned, and what the cognitive revolution provided us with was the brain is a computer and there's not a hell of a lot you can do with that. So this is my aim. I have never thought of it, with all due respect, as disruptive. It just seemed to me to be work that needed to be done. And I was lucky enough to be in a position to do some of that work. Now, so we go first to I will give you my take-home message up front so that you can see at the end how well you think I've done my job here, because we only really develop and grow and improve ourselves if people throw rocks. So I'm interested in rock throwers as much as I am in people who are friendly to my ideas. So the first take home is that, and this is my belief, I think I'm helping to make a case for that. It's not going to be all done today, but I believe that there is a basis for connecting the dots in the evolution of biological cognition up to the emergence of nervous systems. And I think that this basis is both functional and mechanistic, and I've been really, really fascinated to hear in multiple of the talks today talk about function as distinct from mechanism, because it seems like the functions flow through, whereas the mechanisms may flow through in certain instances, but probably are going to be very, very different. The implementation is going to be very, very different in different species. So first to the book. It was written by Peter Sterling and Simon Laughlin, both of whom are scientists in their pioneering scientists in their own right before this, and both of them working in roughly the area of the physiological toll or the metabolic cost of maintaining life, and particularly in the face of stress. So when I came across this book, which had been recommended to me a long time before I ever opened it, I was attempting to develop a Darwinian approach, a biology-first approach to cognition with no a priori commitments. And the most important step in that process was a workshop that was held at the Konrad Lorenz Institute in 2018 that was organized by my dear friend Fred Kaiser and myself, called The Ground Floor of Cognition from Microbes to Plants and Animals and Everything in Between. That marked the birth of what you've heard called basal cognition as an approach, which is basically has 4 principles. First of all, fear not, because people are going to throw rocks, but so what? Start simple, identify principles, and scale up. Now, most of you are going to recognize that this is basic biological practice, but it has not generally been the mode of action in the cognitive sciences for at least not since the early 20th century, and that's actually quite horrifying. A number of midwives of the basal cognition approach are here at this conference, both in person and remotely, and all of them contributed to the special double issue that has been referenced by others. The thing that really attracted me to principles of neural design, because I am not a fan of engineering approaches to biology necessarily, was why does an animal need a brain? It's the second chapter. It's what I refer to as the bacterial chapter, although there are 3 examples of simple systems, E. coli, paramecium, and C. elegans. And the thing that attracted me was the statement on the first page of that chapter that said the brain's purposes reduce to regulating the internal milieu and helping the organism to survive and reproduce. All complex behavior and mental experience, work and play, music and art, politics and prayer are but strategies to accomplish these functions. Now, that's fairly controversial, but I think that they were on to something. And it was also very resonant, congenial to a definition that I've been working on for a very long time as a working definition, a kind of an operationalizable definition of cognition, which the form that it took in the introductory essay to the special issue is: cognition comprises the sensory and other information processing mechanisms an organism has for becoming familiar with, valuing, and interacting productively with features of its environment in order to meet basic existential needs, the most basic of which are survival or persistence, growth and thriving, and reproduction. So from the principles of neural design, any of you who are familiar with the book know that Peter and Simon did not do us the favor of compiling a chart, that these are things that have to be sort of like wrested from the text and refined in whatever ways you can. And these are the core principles that I have derived from their discussion of unicellulars. Now, having gone back again and again, I now see that they have alluded to principles that are beyond unicellulars, but they don't develop them there at all. Okay, so the first one is compute at the cheapest level. Compute with chemistry wherever possible. Now, the principles they're setting out are basically energetic constraints on on the evolution of various functions, mechanisms, etc. That means computing as much as possible within a single cell, sending only what's needed, and sending information at the slowest acceptable rate. The second one is make components irreducibly small. Protein networks operate near thermodynamic, near the thermodynamic limit of energy efficiency, so it's working. The third one is to concentrate resources or sparsify, compute as much as possible within a single cell, send only what's needed, and send information at the slowest acceptable rate. And the final fourth from this section is compute directly with analog primitives, proteins and molecular modulators. Now, thanks to Anita's wonderful presentation on the computation within a single cell, and Allison's wonderful presentation just now, I am not going to have to explain, also in Colleen, sorry to have missed that, I really loved it. I don't have to say much about signal transduction because you have already heard from really, really expert people, and I would only make things much more muddied than they might otherwise be. So the only thing that I will do here is for those of us like myself who are not computationalists and who don't work with dynamical systems, this is just a very, very quick blog on protein signaling for dummies, and it's a simple reminder of a much more complex story. The first point is that a protein's native conformation, the shape that it sits in naturally without being perturbed, and some of them aren't structured, enables its functioning at or near thermodynamic minimum, and it determines its physical and chemical characteristics and its role in the cellular economy. The second point, which Sterling and Lachlan make a whole lot of, is upon binding, proteins can switch into another stable conformation which may alter its receptivity to further binding positively or negatively, and or catalyze further reactions. Cooperative binding via allostery expands this input-output possibilities considerably. This conformation switching capacity can amplify or terminate a signal, reset the information processing mechanism, and equips a single protein to compute by operating as a finite state machine, which those of you who grew up in classical cognitive science will know is a computational model that describes how each of a finite number of states, only one of which can hold at a given time, is achieved given a specified input, usually characterized in terms of a well-defined sequence of state changes, each triggered by a particular condition, such as the presence or absence of an input or a conjunction of inputs. Now, this is all fine and dandy, but what I was really attracted to was their summary of the bottom line, and that is, in brief, simple chemical circuits are capable of implementing procedures used in analog electronics, namely amplify, oscillate, differentiate, and integrate, as well as taking logs and raising to powers. Chemical circuits support arithmetic operations, add, subtract, multiply, and divide. Small chemical circuits also have the ability to perform more complicated functions. What's left out of this are a number of features of bacteria that were not highlighted in the book. The first one is what is almost exclusively called by Jeffrey Stock at Princeton a nanobrain. And those are large chemoreceptor arrays that were discovered around the turn of this century and are now recognized as a highly conserved architecture in bacteria with flagella for receiving and integrating signals from the surrounding milieu. You can see here, whoops, you can't unless I do this. Maybe not. That's not what I wanted. Okay, forget the light. Those large structures in the photos on the top are those signaling arrays. They are also the fluorescing structures in those cells are also nanobrains. They're composed of several thousand chemoreceptors and receptor proteins and sit at the leading pole of the cell like a brain. And the network activity found there is also found in signal transduction pathways, but not with this sort of dedicated and costly infrastructure. They are also extremely complex. Each of the elements in this array are also complex. They people make a lot of heavy weather about, well, you know, they only really register 5 substances that we know of, and they're only really just controlling flagella, and they're very, very simple. But if you look closely at the picture, each of those dimers has 8 modifiable sites. 8 modifiable sites for 5 substances, and they are all the input from these arrays is somehow integrated. We're not sure how to produce a behavioral output. Okay, what's also left out is network activity enabling prediction. This is an example of a predictive behavior within microbial genetic networks. If the troublesome bacterium finds its way into prawns, which then finds its way into the mouth of a human being, immediately that the bacteria enter the mouth and digestive tract, the temperature rises, and heat shock proteins and other mediators are triggered. Secondly, as the oxygen drops, as the bacterium goes down into the digestive system, it begins to switch to anaerobic respiration. And all of this starts at the moment oxygen levels fall, at the moment the temperature rises. What Togopoulos and colleagues found was that these 2 responses, which had to have been learned at some point, which is why I'm really interested in epigenetics, that they're dissociable. And the ability to decouple suggests temporally paired responses in the wild type are not a trivial combined response to the 2 stressors. Second of all, experimental evolution has indicated that the temporal sequence could be reversed over generations epigenetically by conditioning out of phase. And the authors thought this looked like associative learning, but it's not. It might be association over generations, but that's not standardly what associative learning is considered. Another example is anticipatory regulation discovered in E. coli. I'll just, if you want to take note of the article, I'll just bracket that for now, because I'm not sure how much time I've got. 6? Okay. That's scary. Okay, another thing that's left out are behavior modulators, quorum-sensing molecules that behave like hormones, cytokines, and many neuropeptides. All forms of chemical signaling are represented, autocrine, paracrine, juxtacrine, except endocrine, and they seem to be ubiquitous in the bacterial world, and some strains, some species have multiple quorum sensing systems that are designed to help individual cells coordinate their activities with other. That's profoundly social. Okay, the principles of neural design that are assumed applicable to neurozoans is Principle 5, combine analog and pulsatile processing, use more costly electrical signaling for speed over distance, minimize wire through network activity and oscillations. And 7, specialize, complicate for efficiency through the development of cell types with stereotype components. And Principle 8, which is the last one as far as I can tell, is adapt, match, learn, and forget. The combined analog and pulsatile processing is one of my favorites because a bacterial ion channel led to the Nobel Prize for the structure of the ion channel, confirming and outline Hodgkin and Huxley's proposed model. And it didn't bother anybody at all that nobody knew what it was doing in bacteria. And then in 1991, ion channels in Bacillus subtilis biofilms were shown to have the same function as in neurons, to signal rapidly over long distances and large numbers of cells. One of the members of our audience, Jordi Ovajo, is one of the people who developed this, along with his colleague Gurel Suel, and together they are pioneering work in this area that's just mind-blowingly wonderful, that uses mathematical modeling with genetic knockouts and behavioral experiments that are just pushing back boundaries left, right, and center. Okay, so B subtilis combines analog and pulsatile processing. And fun fact, the leading model for investigating the mechanism of anesthesia is a bacterial voltage-gated sodium channel that was discovered in 2007. And now everybody who's interested in the mysterious action of anesthesia is growing bacteria and trying to find out how it works. But is anybody going to ask what it's doing in a bacterium? Are we going to have to wait another 2 decades for anybody to bother? Okay, here's how electrical signaling works in a biofilm. Here is a Bacillus subtilis biofilm with the periphery of the biofilm happily eating. All of a sudden, the cells in the middle start getting starved, and they send out a signal, which is a metabolic trigger in a positive feedback loop that induces potassium release, leading to depolarization of neighboring cells, which then starts to control the status of cells in the interior and the periphery by getting the peripheral cells to stop eating and letting nutrients diffuse into the interior. And then this mechanism is also, they found in subsequent experiments, is also how biofilms can sort of like timeshare on nutrient use between themselves and other biofilms. Another sort of very striking, now this is not memory in the bacterium, but it is an indicator that a biofilm, a Bacillus subtilis biofilm exposed to light with an overlay of a sort of like a screen print can produce a light-imprinted pattern in the biofilm, which is robust and persistent over hours through and goes through various changes through the natural fluctuations in potassium. Another thing that's missing on the minimized wires front, bacteria use extracellular vesicles, which we all know are really, really important in the activity of synapses, to ferry around cargoes that include nucleic acids, toxins, lipoproteins, and enzymes key to microbial physiology and pathogenesis. Another thing that is an important mechanism for minimizing wire and coordinating activity of large populations of cells is oscillations. And here we have a picture of a population of Myxococcus xanthus. The rippling you see is called pretotaxis, and it is formed by the cells moving within and on top of one another and constantly switching direction through the migration of a cluster of proteins. And on the complicate for efficiency front, the UCSD UPF group also discovered that Bacillus subtilis biofilms have a segmentation clock in which their nitrogen stress response is organized in concentric rings that structure spores, and that this patterning is driven by cell autonomous oscillations. And this kind of clock and wavefront process was thought previously to be exclusive to plants and animals, and that phrase that I wish I had $1 for every time I read it. Here's a map of some different cell types in 3 different types of bacteria, and I think I'm going to have to leave you, I'm going to have to bracket associative learning in amoeba, which I'm hoping Jeremy will say something about, but he may or may not. But as far as archaea are concerned, that's totally unknown country. Nobody's doing any memory research that I know of. In bacteria, habituation, sensitization have long been known. At the time that it was identified, people weren't humming and hawing about what those terms actually meant. It just gave the neuroscientists apoplexy, and that was it. And there has been one long-term memory experiment in which B. subtilis were found to hold this memory of a previous exposure for up to 36 hours. So that's that. So the take-home is design principles that inform brain evolution were involved in cognitive function long before nervous systems evolved, and that this alignment connects cognition in bacteria, which, as I'm saying, nobody has to agree with that, with cognition in animals with nervous systems, and supports the notion of a continuum of cognitive function from simple to complex. Thank you very much, and thank you to my team, my funding body, without whose care and support this work would not be possible. Thank you.